It's my pleasure to talk about our research by using plasminogen to treat against COVID-19. My name is Jinan Li. I'm from Talengen Institute of Life Sciences, Shenzhen, China. So first, I would like to introduce more of the background information. As everyone knows, that COVID-19 is causing significant morbidity and mortality. As of February 5th, there are more than 100 million cases have been infected and more than 2 million dead in the world. And this causes unprecedented COVID-19 hardships, both for direct medical and economic impacts, personally and to the family and socially. So there is an urgent need for effective treatment against COVID-19. Currently, the treatment or prevention methods include different types of vaccines, and also a few companies are developing neutralizing antibodies. So next slide, I would like to share some key pathological features of COVID-19. So one of the key features of COVID-19 is the acute respiratory distress syndrome, ARDS. Typical symptoms of ARDS include the formation of hyaline membrane. Hyaline membrane consists of fibrin matrix, serum proteins, and cellular debris, which form a barrier to inhibit the gas exchange and the pulmonary surfactant. Another typical characteristic of the ARDS is the massive diffuse alveolar damage. The second typical features of COVID-19 is the formation of coagulopathy. Nearly 3/4 of patients with COVID-19, they present a hypercoagulable state with the elevated D-dimer and the fibrinogen in the plasma and also other tissues and body fluids. And also there are extensive micro and macro thromboemboli formed in different parts of the body. The third typical key pathological features of COVID-19 is the multiple organ failure. This multiple organ failure is formed by the virus infection at different located organs, and also by the hypoxemia or hypoxia caused due to the ARDS from different part of the body, from the lung. And thirdly is the extensive inflammation, and sometimes even lead to the cytokine storm in the body. Here I would like to introduce briefly about the plasminogen and the plasminogen activator system, PA system, that the plasminogen is one of the key members in. Plasminogen is the precursor of plasmin, which is important, which is a potent ECM protease and important in degrading main components of the extracellular matrix, including fibrin. As almost a classical doctrine, plasminogen is regarded as inert and excessive in the body, and the regulation of the PA system is by the activators and the inhibitors of the system. However, we have worked a while in this system, and we have shown before that plasminogen itself is a key regulator in many pathological processes, including fibrinolysis, wound healing, and infection. So to the left is a schematic presentation of the PA system and its regulation. So the activator, two activator, Tpa or UPA, they are formed by specific cells upon injury, which is also regulated by hormones, growth factors, and cytokines, so on. In the extracellular space, the activities of PAs and the plasmin are controlled by specific inhibitors such as Pi1, which inhibits Tpa and UPA, or Pi2, which more specifically inhibiting UPA. And also the formed active enzyme plasmin is quickly inhibited by alpha-2 antiplasmin through, in case they are not utilized immediately. And plasmin is formed from plasminogen. So plasmin and further, they can degrade extracellular matrix components, including fibrin, or they further activate different pro-matrix metalloproteases to form the active MMP. Together with plasmin, MMP and plasmin, they degrade most parts of the ECM components. So this slide shows our thoughts behind the possible roles of plasminogen in COVID-19. Based on our previous studies and the pathogenesis of COVID-19, we postulate that plasminogen could ameliorate ARDS. That is due to the active form of plasminogen, plasmin, can effectively dissolve fibrin and thus inhibit or break down the formation of hyaline membrane and thus improve the hypoxia, which is to increase the oxygen saturation. The evidence behind this thought is there are studies that as early as 1973 from Ambrose that have shown with the paper titled Plasminogen in the Prevention of Hyaline Membrane Disease. In this study, they showed that intravenous supplementation of plasminogen was effective in lowering the occurrence of premature infant ARDS and death. Further, Hardway and colleagues have shown that treatment of severe acute respiratory distress syndrome, a final report on a phase 1 study, which was published in 2001, that showed that the administration of either urokinase and streptokinase to patients with the terminal ARDS that reduced the expected mortality from 100% to 70%. Further, which was largely from our research team's work, that showed that could improve the coagulopathy and regulate inflammation and also repair different organ damage. So therefore, based on these potential roles of plasminogen, we initiated a clinical study to investigate the treatment efficacy of plasminogen on COVID-19 patients. So the second part of my talk is our research and the findings. So we published one paper called Plasminogen Improves Lung Lesions and Hypoxemia in Patients with COVID-19. This was on the first published preprint platform ResearchGate and SSRN as early as 6th of March 2020, and later in April got peer-reviewed and accepted by QJM from Oxford Press and published on August last year. So in this study, we have tested using plasminogen in 13 COVID-19 patients. This slide describes the treatment profiles of the treatment. So this clinical study was performed during the 10th to 20th of February 2020 at 2 hospitals in Hubei province. The province where Wuhan, the outbreak city of COVID-19 was, the nearby two hospitals. So in this study, we have used the sterile freeze-dried plasminogen of 5 milligrams per vial, produced at facilities with GMP compliance and given by optimization inhalation. The safety of the reagent, particularly the virus activation process, was confirmed by a certified third-party testing organization and also by governmental authority. This plasminogen was originated from the plasma. So to the left is the treatment profile of the patients. So we included 13 patients, both males and females. Their age range as young as 30 to as late as about 78 years old, and severity of the disease range from moderate, severe, and also critical. So these categories was based on the governmental criteria. So we have made the plasminogen inhalation either as little as 1 time or as much as 6 times, depending on the facilities, because these patients it was very difficult to apply these reagents to under such rush conditions. So this slide shows the plasminogen improves lung lesions of clinically moderate COVID-19 patients. So to the left is the high-resolution CT images of 5 moderate COVID-19 patients with different IDs, and 8 columns shows their chest CT images before the plasminogen inhalation, and B column shows the CT image after the plasminogen inhalation. And for one patient, we had the chance to check the CT image of the chest 5 days after the last treatment. And then the black arrows, it was difficult to see here, but here it exists, and here all boxes, they particularly point the abnormalities. So despite the 5 patients that received the daily common supportive treatments, the density and range of the ground glass opacities in lungs were increased over time, that suggests a rapid deleterious progress of the disease. Among the 5 patients, number 3 was the relatively mildest. So the CT results before the first inhalation, patient 3, it showed the multiple ground glass appearance in the lung characterized by unclear margin and heterogeneous density. After the last treatment, the number and area of lesion focal decreased and almost all disappeared for this patient. The other 4 patients with relatively more severe conditions, the CT image also showed the multiple ground glass opacities in the lungs characterized with unclear margin and heterogeneous density, especially they have showed some consolidations. So after the treatment, CT results showed the number and range of the lesion focal decreased and partially disappeared. It seems like the outcome results somehow correlate to the original severity. So for patient 5, particularly the lowest one, interestingly, even 5 days after the treatment was withdrawn, the improvement still continued as compared to a few days before. Next slide, we show that plasminogen improves oxygen saturation of clinically severe and critical conditioned COVID-19 patients. So this table shows the oxygen saturation values of these 8 patients. 6 were severe and 2 were critical. They are in the ICU ward. So their oxygen saturation values before the first treatment will range from as low as 79 to 82% for the critical ones, or to somehow at the border of the severe condition category. In China, it was regarded as severe if the oxygen level was below 93%. So after the different times of treatment of plasminogen, in general, their oxygen level have all increased. For this, only one that did not increase much was because it's only used once. Particularly for these 2 critical patients, their oxygen levels have significantly increased from 79 to 92% to 91% just within 1 hour after the first inhalation and stabilized at around 91% thereafter. So the quick improvements of the oxygen saturation levels, particularly in the critical condition, the patients, somehow also suggest that using plasminogen is an effective way to quickly alleviate these patients. Further, we show that plasminogen improved heartbeats of COVID-19 patients. So for all these 13 patients from moderate, severe to critical conditions, their heart rates before the treatment was in general higher than ordinary, but after treatment, in general, the heartbeats have all decreased. Although very limited patients, but if we group them to these 3 severity groups and did somehow comparison, we could see probably because of the number of the patients that we saw that in the moderate group, the degrees of the heart stress was statistically significant. And even though these critical condition patients were only 2, but we did observe a sharp decrease of the heart stress, suggesting if we could have the chance to have more patients included, we could observe a more clear relief of heartbeats in the critically severe patients. So to summarize our research, we have found that plasminogen seems to improve the lung lesions of clinically moderate COVID-19 patients, and plasminogen improves oxygen saturation of clinically severe and critical COVID-19 patients. And also plasminogen improves heart rates of these patients. Therefore, additional plasminogen by atomization inhalation seems effective, efficient, and relatively easy in treating lung lesions and hypoxemia caused by COVID-19 infections. This study, we believe, highlights a hope of efficiently combating this rapid epidemic emergency. So next, we would like to show the summary of other researches after our publication. As I mentioned before, we used the plasminogen, which is not an approved drug by the FDA yet, as early as February of 2020. So probably we are among the first of using novel drug candidates to treat COVID-19, and for sure, the first one in the first drug candidate in the plasminogen activator system. So after that, we have gradually observed the studies on the members of the PA system, and also using particularly TPA, which is approved drug on the treating of these patients. So some those results have confirmed our predictions. So this slide shows that the high levels of Tpa and Pi1 in patients with COVID-19. So for two papers, just example for two papers, from two groups, they have shown that plasma Tpa and the Pi1, they are in hospitalized COVID-19 patients, or hypofibrinolytic state and high thrombin generations play a major role in COVID-19 associated thrombosis. As you can see from to the left, both Tpa and Pi1, they are extremely higher in the COVID-19 group as compared to the control ones. And their Pi1 and Tpa level seems to be correlated. To the right is the laboratory findings of the COVID-19 patients between ICU group and the non-ICU group. And again, it shows that both Tpa and Pi1, they are much higher in the ICU groups compared to the non-ICU one. Then what we try to summarize based on our understanding under current results available, the advantages and disadvantages of TPA in patients with COVID-19. As I said before, TPA, which is the main physiological plasminogen activator in the circulation, it's a fibrin-specific thrombolytic agent that's been approved for the treatment of acute thromboembolic diseases. So hence, theoretically, TPA would reduce the sequelae of thromboembolic disease associated with COVID-19. When we put the advantages analyze advantages of TPA, TPA is very fibrin specific, and it has rapid clot lysis functions, although it only dissolves fresh thrombus, and it's an approved drug for thrombolysis in the area of acute ischemic stroke and acute myocardial infarctions. But also, TPA has its disadvantages, such as highly limited therapeutic time window for dissolving clot. It's it's known if it's based on evidence that probably within 4.5 hours after the clot formed, it's efficient in dissolve the fresh clot. It has a short half-life. For the natural ones, it's only 2 to 6 minutes, although in the new mutant ones could go up to 130 minutes. It has a dose-dependent manner in dissolving fibrin, but also it could have severe side effects such as hemorrhage and nerve toxicity. And so far, its functions is restricted to fibrinolysis. So therefore, TPA may be useful in treating COVID-19, but probably accompanied by a lot of side effects. Sometimes, as we know, that could even leading to death. Here it shows one such paper. So as I said, we try to put together the limitations of TPA in patients with COVID-19, particularly. We postulate that it seems it can't decrease the mortality of COVID-19 when we try to summarize other studies. As we know, the overall mortality of COVID-19 is between 1 to 5%. However, if these patients, they are having ARDS, COVID-19 with ARDS, then the mortality quickly increase to 22 to 64%, depending on the different studies. When we did a little literature search and summarized the 5 case reports about TPA application in COVID-19, we found that 7 out of the 14 patients that used in these 5 studies that had been administered with TPA, they died. That means the mortality of COVID-19 with TPA is maybe 50%. So if you think the mortality of COVID-19 with ARDS is from 22 to 64%. Thus, it seems like TPA does not decrease the mortality or rescue the patients, particularly with ARDS. Also, other studies which I will have shown that it has limited efficacy, as we shown before, that it has those patients have a very high Pi1 level, which inhibits TPA. And these patients, they already have very high TPA level already. Then you would wonder why adding more, even more TPA. And these patients have low plasminogen level, which I will show next. And as we have described, that the COVID-19 is not just thrombosis related. So this slide further shows the circulating plasminogen concentration in COVID-19 patients. So if you compare to the COVID-19 positive and sick controls, they have similar level of plasminogen. However, if you group them between the hospitalized ones or the discharged ones or recovered ones, you see that they are the discharged one had significantly increased level compared to the hospitalized ones. The hospitalized ones, they have same plasminogen levels as compared to the normal sick ones. Similarly, if they are not severe as in the ICU group, then their plasminogen level also increased. So this is very interesting. That shows somehow, because of the defects of naturally increasing plasminogen, they could cause the fate of this patient, either hospitalized or ICU'd. So here we put together the advantages and disadvantages of plasminogen in patients with COVID-19. Advantages we try to put together, they have key roles in many pathological processes, including fibrinolysis, wound healing, infection, inflammation, as we have studied. And also it improves multiple complications or comorbidities, such as hypertension, diabetes, cardiovascular diseases, and tissue organ damages. Dissolve acute and chronic thrombus. It has specific targets the injury site. So rather than TPA extensively activate the fibrinolysis process. And it has high safety profile. We have observed, and also from literature, there's no hemorrhage observed, not observed nerve toxicity or any other related adverse reactions. And it has a long half-life from 19 to 53 hours, and it has broad therapeutic time window ranging from early, middle, late stage of the healing. The key disadvantages is not approved yet as an agent of treating COVID-19 or even any diseases, which we are trying to work on. So above all, it seems like the plasminogen may be an effective and efficient way to treat COVID-19 and associated complications. So here listed some of our papers and also our patents surrounding about symptoms of COVID-19. So finally, it's soon 30 minutes. I would like to acknowledge all our staff involved in this study for their assistance, including the authors of the paper, but also other staff that from these 4 organizations, Talengen Institute, Chang'an Hospital, and the 2 hospitals from Hubei province. Thank you for your attention.